Welcome to this lecture on Introduction to Complex Biological Systems. So, this week I am teaching Evolution, the History of Life, and today in this lecture I am going to talk about evolution of eyes. Eyes or eye is a fantastic organ, and there are all these different types of eyes that we see in nature. So, this is eye of a spider, an elephant, eye of a gorilla, owl, goat, octopus, human. We are going to compare the eyes of human and octopus in details later on. Cat, beluga, so it's a whale-like fish, donkey, mosquito, Drosophila, mantis and lobster. So, I am also going to discuss these eyes, dragonfly and mosquito eyes. So, these are compound eyes. So, it turns out that this organ accomplishes vision. However, if you look at this, all of these look so different, which means that nature has come up with the same solution in multiple different pathways. So, you can consider that these are examples of convergent evolution. So, as we have seen that Earth was formed 4.5 billion years ago, right? And sunlight turns out to be the most potent force for controlling the evolution of living organism, because sunlight is the energy source and we are trapping this energy to perform all the work that we do, anything that goes on this planet. Evolution of eyes is also the consequence of light on Earth. Because organisms use light to see, eyes have evolved into different shapes, sizes and design. Within this light, there are light sensitive neurons and proteins responsible for light sensitivity. So, I will discuss the architecture of our eye in details in the next few slides. Eyes have evolved from simple light sensitive eye spot to lens containing complex eyes. Eyes have evolved independently in different organisms. So, in the previous slide, I showed you all the different types of eyes or different looking eyes in these different organisms. So, they have evolved independently of each other. Various factors behind the evolution of eyes are like proteins. So, evolution of proteins, for example, opsin will drive a design of an eye. Environmental factors, for example, animals that live on land versus animals that live in water, their environmental factors are different. So, that will also drive their evolution. Protection from UV, color filtering and genetic factors like the Hox genes. So, all of these together will they have impacted evolution of eye, and that is why we see so many different designs. In fact, if we talk about evolution, it turns out that there are three major factors that drive evolution. The first one is utilization of energy, right? So, every organism needs to find a source of energy for survival, and that energy we get in the form of consumption of food, right? So, search of food is something that drives evolution. An organism which has a better eye will of course be able to locate food better and hence will have a better chance of survival. So, search of food will drive the evolution of eye. Second is the predator-prey relationship. So, every organism is a predator, so it eats something, and it can also be a prey because it can be eaten by something. So, as a predator, an organism has to locate its prey, and as a prey, that organism has to evade its predator, right? And all of these things can be done better if your vision is good. So, this predator-prey relationship will also drive evolution of eye. The third factor is reproduction. So, for higher organisms, an organism has to find its mate, right? So, here also vision plays a very important role. So, energy source, predator-prey relationship and reproduction, all of these three things together drive evolution, and the evolution of eyes have also been driven by these three factors. So, here is the anatomy of the human eye. So, this is the human eye, and the major portions of the human eye are shown. So, we have a lens, there is this retina. So, the lens, it concentrates the light and it the light falls on the retina and then image is formed. That image is converted into electrical signal, and that electrical signal is conducted by these optical nerves to our brain, and that is how we perceive or we see something, right. Now, lens is also made up of proteins, right, but its function is to put the light in here, right. So, there is a pinhole results in sharper images. Lens will bend all the lights and concentrate them on the retina, and this is the architecture of the retina. So, our retina is primarily made up of neuronal cells. So, there are two types of neuronal cells, rod shaped cells and cone shaped cells. Okay. So, the lens focuses light on the retina, which is composed of layers of light sensitive neurons. The primary photosensory neurons are rod cells and cone cells. So, it turns out that there are 10 to the power 9 rod cells per retina and 3 times 10 to the power 6 cone cells per retina. Rod cells are responsible for high resolution images and night vision. However, these rod cells, they cannot discriminate between colors. Okay. So, they give you high resolution image and they can also help you see in dark. Okay. So, night vision. The cone cells, they can distinguish between the 3 primary colors. Okay. So, the color that we see are because of the cone cells. So, they initiate color vision. Rods and cones form synapses with several ranks of interconnecting neurons that convey and integrate the electrical signals. The signal eventually pass from the ganglion neurons through the optic nerve to the brain. So, these rod cells and cone cells, they will connect through these interconnecting neurons, these green ones, and then these ganglions, right, and then these are the optical nerves which go to the brain. So, this is an amplified image of what you see here, right. Now, there is something very interesting that I should point out, and I will again discuss this in more details later on. The design of this is inverted. Inverted in the sense that these are the cells which sense the light. However, light is coming from this direction. Okay. So, light is coming from this direction. So, it passes through these optical nerves and all these neurons, and then it will reach the rod and cone cells. It would have been better if the rod and cone cells were on this side and these other cells would have been on the other side. And we will see that is exactly the case for organisms like octopus, right. So, it means that the eyes so we will compare eyes of human and octopus and we will see that they are very similar. However, this particular arrangement is inverted, and this inversion makes the octopus eye much better than the human eye. Okay. And another fact that shows up here is you see these optical nerves which are coming out here. These optical nerves, because of these optical nerves, there are no neuronal, this rod and cone cells in this particular region. So, that particular region creates a blind spot. So, our retina has a blind spot where if light falls, no image will be created, and that blind spot is absent in the eye design of octopuses. Okay. So, let us look at these rod cells and cone cells in more details. So, this is a rod cell, and it is divided into 2 segments. One is called the outer segment and the other one is called the inner segment. So, this is the outer segment and this is the inner segment. And in these segments, there are all these ion channels, right. So, the outer segment is filled with stacks of membranous discs containing the photoreceptor rhodopsin. So, rhodopsin is present in this outer segment. I have already talked about rhodopsin and I will discuss this again in today's lecture. The inner segment contains nucleus and other organelles. So, nucleus and mitochondria and all those organelles are present in the inner segment. So, this is the working part of this cell or the neuron. Membrane potential is reduced. So, there are these ion channels. So, this is sodium potassium ATPase. So, what it does is it hydrolyzes ATP and pumps out 3 sodium ions and pumps in 2 potassium ions. So, the net charge difference is +1 on this side. So, it pumps in 2 potassium ions and pumps out 3 sodium ions, right. Now, the sodium ions which are in higher concentration here, they flow in through these sodium calcium ion channels. So, these sodium calcium ion channels, these are open. These are in this open state and they will allow the flow in of this sodium and calcium ions. So, this equilibrium results in a voltage difference of 45 millivolt across the membrane. Now, this ion channel is open when cyclic GMP binds to this ion channel. So, it turns out that roughly 3 cyclic GMPs need to bind this ion channel for it to be completely open. When light falls on this cell, it will fall on, so it will be detected by the rhodopsin. So, that is another protein which is in this part. Okay. So, rhodopsin, it will absorb the light and it triggers a reaction where this cyclic GMP will be degraded. Okay. So, it will be hydrolyzed, resulting in drop in concentration of the cyclic GMP amount. So, if the cyclic GMP amount drops, then it will not bind to this channel and this channel will be closed. So, without continuous influx through this channel, because the sodium and calcium ions were going in, so these positive ions are not going in, but they are still being pumped out, there will be a charge difference. So, the membrane becomes hyperpolarized and this electric signal is passed to the brain through the ranks of the neuron that we saw in the previous slide. So, a potential is generated by this and that will be passed on to these neurons and finally through the optic nerve to the brain. So, light is converted to an electrical signal by rhodopsin and these ion channels. So, this is rhodopsin, we have already seen this, it is a membrane protein, it has these 7 transmembrane helices which is embedded in the membrane. And almost at the center of this protein is this particular molecule which is retinol, and we have seen that it comes from vitamin A. Now, retinol that is bound in rhodopsin has this cis conformation. So, between the 11 and 12 carbon, this is in the cis conformation. When light falls, this converts to trans retinol. Okay. So, it converts into the trans conformation and that results in this big conformational change in one of the helices. So, you see that there is a 7 angstrom shift of this helix and a 2 angstrom shift of another helix. So, this conformational change triggers a series of biochemical reactions, right, which will result in what we have discussed in the previous slide. So, let us see what happens here. So, this is the vision cycle. I have already discussed this in one of the previous lectures, but here I am going to discuss it in more details. So, one half of the cycle is the excitation cycle and the other half of the cycle is the recovery or the adaptation cycle. So, this is rhodopsin, it is bound to the membrane and this is retinol, cis retinol. When light falls, the cis retinol gets converted to trans retinol and this results in a tug on or pull on one of the helices. So, there is a conformational change and that conformational change activates this other protein. So, activated rhodopsin catalyzes replacement of GDP by GTP on transduction. So, this protein is transducin. Change in conformation of rhodopsin results in kicking out of GDP from transducin and it binds to GTP. So, transducin now binds to GTP, right, and then it comes out from here and it goes and binds to this protein complex. So, this is PDE or phosphodiesterase. Now, this phosphodiesterase is originally in an inhibited state. So, this I is an inhibitor which is inhibiting this phosphodiesterase. This GTP bound transducin comes and it binds to this inhibitor more tightly than PDE, which means that it will take this out like so. So, now the phosphodiesterase is free and it is activated, and what it will do? It will hydrolyze this cyclic GMP. So, it will break one of the phosphodiesterase bond, So, it will reduce the concentration of cyclic GMP. as the concentration of cyclic GMP goes down, this gated ion channel, which was allowing sodium and calcium ions in, this becomes closed because cyclic GMP concentration has gone down. No cyclic GMP binds here, so this ion channel closes like this. So, this influx is stopped, right, and that results in the generation of this electrical potential, which is roughly 1 millivolt, and it is carried on through other neuronal cells and the optical nerve into the brain. So, this is a continuous efflux of calcium through the sodium calcium exchanger, right. So, this is still happening. So, this is still happening, but no calcium is coming in, but calcium is still going out, which means that calcium ion concentration also decreases. So, we have already generated the potential and that is transmitted to the brain. Now, we are in the recovery cycle. So, lower calcium concentration triggers, so this reduced calcium concentration activates this enzyme, which is guanyl cyclase, this enzyme GC. So, guanyl cyclase gets activated because of low calcium ion concentration. It inhibits PDE, which is this. So, it inhibits PDE. So, it will stop the hydrolysis of cyclic GMP, reopening the cation channel and results in this level. So, it will inhibit PDE and it will synthesize more. So, it will use GTP to synthesize cyclic GMP. So, in essence, it stops this and it makes more cyclic GMP. So, it will increase the level of cyclic GMP, and those cyclic GMP can now go and bind to this ion channel, opening it. So, the ion channels which were closed now, they will get opened, and now again sodium and calcium ions will rush in through this ion channel, right. So, this raises towards the dark side. Okay. So, so now we are going to the equilibrium. Something also happens to the rhodopsin. Now, see that rhodopsin is bound to the transretinol, right. So, rhodopsin kinase phosphorylates rhodopsin. Okay. There is already low calcium ion at this point. So, this low calcium ion and recoverin, this is another protein, it stimulates this reaction. So, it phosphorylates rhodopsin. Arrestin binds the phosphorylated carboxyl terminus and inactivates rhodopsin. So, arrestin, this protein, and recoverin, they together come and inactivate the rhodopsin. At this point, arrestin slowly dissociates, rhodopsin is also dephosphorylated. This transretinol is now replaced by another cisretinol, okay, because transretinol has to go out, cisretinol has to come. Now, this cisretinol is present, so it goes back to this state. Calcium concentration has recovered because these ion channels have opened. Cyclic GMP levels are also back up. So, we are back to this condition. Another photon comes and then the whole cycle will get repeated. So, this is the vision cycle. So, in essence what happens is that the light energy or the photon is converted to electrical signal via these enzymes. Now, it turns out that one rhodopsin can trigger the activity of a lot of this protein, which is transducin and the phosphodiesterase, and they will catalyze a lot of cyclic GMP hydrolysis. So, the signal is amplified. So, amplification of the visual signal by this rod and cone cells. So, each excited rhodopsin can activate 500 molecules of transducin. So, each activated rhodopsin can activate almost 500 molecules of transducin. Each transducin will activate one phosphodiesterases. So, one rhodopsin in turn activates almost 500 phosphodiesterases. So, resulting in the activation of 500 phosphodiesterase. Now, one phosphodiesterase hydrolyzes almost 4200 cyclic GMP per second. So, 500 will hydrolyze this into 500 per second. Okay. Now, we have seen that 3 cyclic GMP binding is needed to open one of this cyclic GMP gated cation channel. So, if we have hydrolyzed so many cyclic GMPs, then the cyclic GMP concentration goes down very rapidly and almost 1,000 such ion channels are closed. So, one photon results in the closing of 1,000 or more of this cyclic GMP gated cation channel, right, which results in a large buildup of ions and results in a potential difference of almost 1 millivolt. So, there is a large signal amplification because of this biochemical reactions. So, one photon results in this electrical signal. Okay. So, that was the human eye, right. But it turns out that there are many designs of eye and some of those are very primitive, but we do see examples of this primitive eye architecture even today. So, one is this simple light sensitive cells. Okay. So, there is no architecture, only these light sensitive cells. When light falls on it, that will be detected. So, they can detect light versus dark. A little better design is where the cells are cup shaped. In this case, again no image is formed, but it can sense light and dark and also some sort of directionality, like from which direction the light is coming. Here the design is where it has a pinhole, but there is no lens. So, this is filled with water. So, if it is an aquatic animal like nautilus, it will be filled with water. So, here it will not form an image, but it can see a shadow of something passing by. So, it can sort of see in which direction a particular prey or predator is coming or going. This is a primitive eye with lens and this is a complex eye with a very sophisticated lens and much more complex design. So, this will be something that we have. So, light sensitive cells. Simple light sensitive cells in primitive unicellular organisms have a primitive eye spot that helps them detect light and move towards it. So, only thing that these cells do is that they detect where the light intensity is more and the organism will move towards that. So, that will help in better photosynthesis. So, it optimizes sunlight for photosynthesis. These light sensitive cells have light sensitive proteins known as opsins. So, rhodopsin will be an improved or more optimized version of these opsins. Opsins emerged in unicellular organisms over 600 million years ago. These opsins combined with other light sensitive molecules to enable organisms detect light direction, darkness and intensity of light. So, one example of a single celled organism which has this light sensitive cells, one example of an organism which has this light sensitive cells is Euglena. The eye spot in Euglena is a red organelle made of carotenoid pigment granules. So, we have already seen carotenoid structure in one of the previous lectures. So, when there is light, it moves towards the light source to do enhanced photosynthesis. When light reaches the eye spot of Euglena, the photoreceptor proteins present change shape. These changes alter the structure of the flagella and influence Euglena's movement. So, it has flagella here. So, they will start spinning and it will push this organism towards the light source. So, this movement as a function of light is called phototaxis. So, photo light and taxis means movement. Okay. So, we have seen this. Now, eye cup. Okay. So, cup shaped eyes. The formation of cup shaped eyes provides an evolutionary significant event where flat light sensitive cells evolved into better curved structures. So, instead of a flat structure like this, it becomes a curved structure like this. This shallow depression allowed the light sensitive cells to detect the direction of light. So, in this case when light falls, it still detects it, but it is not doing a very good job, but here it can detect the direction of light. So, if light is coming from this direction, there will be more illumination of this, versus if light comes from this direction, there will be more illumination on this. So, based on that, it can detect the direction of lights better than these flat cells. Light reaches the cells through an angle and at the depression depends the direction of light becomes finer. Flatworms have cup shaped eyes. So, that is an example. The cup-shaped eyes detects changes in light intensity, helping in navigation and detecting predators. So, it can detect predators and it can also detect prey. Flatworm cup-shaped eye spot is located at the anterior end and senses light intensity. So, it allows flatworms to orient themselves in the environment by detecting the intensity of the light. The eye spot is not an eye as it lacks a lens and other important structure of an eye that allow correct vision. So, it is not like a modern eye that we have, but it is called an eye spot. So, this is a flatworm and it has eye spots here. So, what you have is this cup shaped eye. So, these are the light sensitive cells and then these neurons, they are the photoreceptors. So, they will transmit the signal. The formation of cup shaped eyes provides various advantages such as ability to detect the direction of light, sense environment changes and enhance the survival of the organism. The cup-shaped eyes do not form images as they are only able to detect light and dark. In the process of evolution of eyes, the development of cup-shaped eyes plays a crucial step towards more complex eyes, indicating important changes and adaptation over time. So, once this happened, you can imagine that addition of more components will lead to the modern complex eyes. So, it is sort of shown here that pigment spot, this is flat and then there are these nerve fibers which carry the signal. Now, we have this cup shaped which is better in detecting the direction of light. In this case, the cup shape is there, but instead of this broad opening, there is a pinhole. So, once there is a pinhole, now better images can form and this starts to become an eye where it can actually start seeing images. The resolution will depend on how sophisticated this design is. So, you have to put a lens to make it a better, make better resolution images. So, cup-shaped eyes provide various advantages, but as the structure depends, it creates a small pinhole aperture. This structure allows light to pass and limits light scattering. This pinhole structure improves the resolution and enables weak image formation. There is no lens, so this is filled with water. So, it will result in the formation of a weak image or a low resolution image. So, Nautilus, they have pinhole eyes, it does not contain a lens. So, this is actually filled with water. The biggest limitation of pinhole eyes is the formation of low resolution images that are dim and lack detailed information. So, this is Nautilus and this is their eye and this is a zoomed image of their eye. So, it forms this pinhole and this cavity, it does not have a lens, however, it is filled with seawater, right. So, you can imagine that it is not going to do a very good job of concentrating the photons and whatever image is generated will not be a very sharp image. The next is eyes with lens. So, these are called lens eyes. Lens is a transparent curved structure for focusing light and providing a clearer vision. Clearer images lead to better depth perception and sensing of the surroundings. So, there are obvious evolutionary advantages of seeing better. Lenses are found in eyes of vertebrates like humans, reptiles, birds, mammals, etc. and cephalopods like octopus, squid, etc. The biggest advantage of lens containing eyes is that lens adjusts its shape to focus on objects at different distances and conditions. Lenses collect the light, bend precisely onto the retina for the image formation. This helps to collect the fine details of the object and understand the surrounding in a 3 dimensions. So, we get much sharper images because of the lens. It turns out that lenses have developed independently in different organisms such as the vertebrates and the cephalopods, and we are going to compare the eye architecture in vertebrates and cephalopods. So, vertebrates and cephalopods both have very sensitive and sophisticated visual systems best suited for their environment, but we will see what are the major differences. And again, this provides an example of a convergent evolution. So, some unique adaptation of lens in eyes, for example, in case of fish, they can analyze changes in the refractive index underwater, providing a clear sense of the environment. Birds, on the other hand, they fly very high, so they have to sense or detect things from a distance. So eagles, they have extremely sharp vision, enabling the detection of prey from very, very far away. Cephalopods, they have high adaptability to low light underwater conditions. So they can go down to at very high depth, so where light reaches very little light reaches. So in these low light conditions, they have ability to see. Chameleons, they have independently moving eyes, which means that the eyes move independent of each other. In our case, both eyes move together, right? So these independently moving eyes provide a broader field of view for spotting prey and predators. Humans, so we can differentiate between different colors. We see high resolution image, which helps us in reading, playing, etc. So again, this is the architecture of the human eye. Now, as I pointed out earlier, there is this weird design where the retina is on the back and the nerves are in the front. So when light comes through, it goes through this thin layer of nerve cells and then reaches the retina. However, at this position where we have these optic nerves, so all of these nerves are bundled together and they go to the brain, there is no retina cells here, right? So no rod cells or cone cells, and this particular region creates a blind spot. So this is the blind spot in our retina. Okay. However, in case of cephalopods, the design is the way you would expect. Retina is in the front and the nerves are in the back. So even though the nerves are going here, retina is there. So even though the nerves are there, since the retina are in the front, there is no blind spot in cephalopods. So vertebrates, we have this inverted architecture where the retina is in the back, nerves are in the front. That results in this blind spot. In case of cephalopods, retina is in the rod and cone cells are in the front, the nerves are in the back, and there is no blind spot. There is this other design of eye which is called the compound eye. So this is Drosophila, and you can see this compound eye in Drosophila. Compound eyes are found in arthropods such as flies, bees, and crustaceans like crabs and lobsters. These compound eyes are composed of multiple repeating units. So you can see this array of lenses, right? These are called omatida. Each of these functions as an individual visual unit having its own lens and photoreceptors. These organisms visualize the world in a mosaic-like manner, which is optimum for motion detection and provide a wide field of vision. So a flower that we see like this, these organisms, they will see it like this. However, this provides much better reaction time for these organisms. Okay, so they can detect motion much better than we do. Compound eyes add various advantages such as wide field of view, so almost 360-degree view, helping spot predators and analyzing the environment, rapid motion detection. So since each of them changes independently in the light, so it provides quick response time. Adaptability, so increasing survival in different habitats like terrestrial to aquatic. Bees have compound eyes that can detect UV patterns on flowers, and this helps them for pollination and navigation. Compound eye provides better motion detection, and it covers broad area vision compared to lens eyes that we have. But complex eyes make lower resolution images compared to lens eyes. So this will be compound eyes. So these compound eyes, they make lower resolution images compared to lens eyes because of this mosaic pattern. Compound eyes are good for smaller organisms that require quick response. However, complex eyes is better for larger organisms like us and other vertebrates. So we have this complex eyes. So complex eyes, these are advanced visual systems. They have structures like lens, retina, and the photoreceptor cells. So examples will be eyes of human, octopus, squid, and insects. Complex eyes provide various advantages over others such as high resolution images, advanced light sensitivity, color vision, and also good motion detection. So, complex eyes demonstrate the ingenuity of nature from simple light sensitive cells to image forming eyes. So, we have seen that all these different designs, there are some very primitive designs, and now we have very complex eye design like this. So, on top of that, there are also some protective membranes. For example, we have eyelids which protects our eyes. We have tears which which produces lysozyme that fights against microbes and protects our eyes from infection. Nictitating membrane, a third eyelid in some animals like birds and amphibians that provides extra protection without obstructing vision, like you see here. So, this shields aquatic animals from water currents and save desert animals from intense and harmful UV radiation. So, these are the different eye type: cup shaped, pinhole, lens eye and compound eyes. So, they are found in these different organisms. Their advantages and disadvantages are listed here. So, you can pause this and go through this. So, this is a summary of the different eye types. So, it is interesting to compare human eyes with octopus eyes. Human eyes, we have a blind spot, as I showed in the previous slide, because of the inverted design of the retina and the nerve cells. No such blind spot is present because the photoreceptor face forward. They are on the front side and the nerve cells are on the back side. This is highly adaptable for terrestrial environments. Octopus eye is also highly adaptable for underwater vision. A broad spectrum of colors, vision and depth perception is done by human eyes. Octopus eye, they can detect polarized light and camouflage easily underwater. So, both of these eye types are examples of convergent evolution. So, I have listed all these different references in the slides, and I have also listed some of the important references here. You can go through them and you can also use the books that I have listed earlier. Thank you.